Thanks, Suzanne and Lou and Kelly. I'm always delighted to be here at NIMBioS. It's a wonderful institute, and Knoxville is actually quite lovely. So I was out earlier this afternoon just walking around looking at the buildings and the shops and the restaurants and things. And if you've never had a chance to visit Knoxville, I highly encourage you to try and get out maybe an evening or something and walk around and see what it's like. So the speakers were given a very challenging task. We were asked in 30 minutes to tell you both about our research and our career trajectories. So I'm going to see if I can accomplish that and not speak so quickly that you don't understand what I'm saying. And so as you know, I'm currently the faculty and associate professor in the School of Mathematics at Georgia Tech. And my research area is what is called discrete mathematical biology. That is, I'm interested basically in the interaction of combinatorics, which is a different area of math than one normally thinks about when one thinks about mathematical biology. So usually with mathematical biology, you're thinking about dynamical systems or differential equations or maybe statistics, but not often about discrete structures. But I'm very interested in structures that look like this and also in structures that look like that. So basically what mathematics can tell us about biology, but also there's other directions. So that is, thinking about biological questions inspires us to think about mathematics in new and interesting ways. And so that really should be this cycle of not only does mathematics apply to biology, but biology should also motivate new and interesting mathematics. And so for the record, I in fact started college as a biology major. So I did 2 years as a biology major because I thought I would want to study biological systems. But in fact then finished with a mathematics degree from the University of Illinois at Urbana-Champaign and went on to do a PhD in math at Berkeley. And so when I was at Berkeley, my advisor was John Rhodes, and he's an expert in semigroups. So as you may recall from algebra, semigroups are defective groups. They have only a set with a product that is associative. And one of sort of the canonical types of semigroups are just combinatorics on words. So basically, you have strings of letters, or you have letters, and you concatenate them to make strings, and you look at the properties that those have. And so it was sort of fairly natural. So my thesis actually had no biology in it whatsoever. It was a question that arises in varieties of semigroups, or equivalently, you can think about it as a generalized pattern matching problem, so there's computational aspects to it, but there was no biology to it. But I went on to do a postdoc at the University of British Columbia, actually in the computer science department. And when I was there, I met a woman named Anne Condon, and she's the one who introduced me to these computational biology questions. And she thought I might be interested in them because there's this underlying sort of strings over a finite alphabet connection. And I said, that sounds interesting. And ironically, I started college as a biology major. So I even have some of the vocabulary that I needed to start working in this area. So going even farther back than your algebra class, to maybe high school biology, although actually in this room you might have been more recent than that. But if you don't normally think about molecular biology questions, then the structures that I work on are actually RNA molecules. And so RNA is like DNA in that it's a nucleotide alphabet over these 4 letters, A, C, G, and U. But it's unlike DNA because most RNA sequences are single-stranded as opposed to the canonical double-stranded DNA helix. And the reason that this is important is because those single-stranded RNA sequences base pair with each other, and those base pairings form structures that perform functions. So DNA structure is well known. I've qualified this but all statements about biology are qualified to the current order of approximation. And again, this is an audience that should probably understand that pretty well, that it's very hard to make definitive statements about biology. But we can say that DNA structure is pretty well known, at least at this level. And of course, this is the famous photo 51 that was taken by Rosalind Franklin and then used by Watson and Crick to infer the double-stranded DNA helix. On the other hand, I say RNA structures are still largely unknown. And the reason for that is because these single-stranded RNA sequences can form many different types of base pairs, and those base pairs occur in complicated ways. So I've been talking about this base pair hybridization. So basically, you want to think about RNA structures in terms of their dimensionality.
Okay? So at the one-dimensional level, we have the primary sequence. That's just the linear, A, C, Gs, and Us. In 3 dimensions, these are molecules that have a structure, and that structure is essential to the function that they perform. So the structure that you see here is actually the tRNA molecule, and that's really important because those 2 ends of the molecule, one of which is pairs with the reads the RNA, and the other end holds on to the amino acid, and that's how that's how proteins are coded from DNA via RNA. But the structure that I work on is actually what's called the secondary structure, and those are this set of nested canonical base pairs, and that's effectively the 2D structure. So it's an intermediate structure between the 1D and the 3D, and it's understood that knowing the 2D structure is a major stepping stone towards understanding the 3-dimensional structure. So ultimately, we'd like to know the 3-dimensional structure, but that's a really hard problem, and so we're going to settle on only a slightly less hard problem, that is knowing the secondary structure. So why do we care about the secondary structure? Well, for the purposes of this talk, we're going to focus on RNA viruses. So many viruses code their genomes in RNA instead of DNA. One example would just be the common cold, or the rhinovirus. And so this is the cover of Science. Anne Palmenberg is an RNA virologist that I met when I did a postdoc at the University of Wisconsin in Madison. I was actually a computational biology postdoc there. And met her and started getting interested in these RNA viral problems. And what you see here is the viral capsid, and then behind it, blown up, are the different base pairs of the secondary structure. And there are examples where knowing the base pairing of the secondary structure has an effect on function. So dengue, for instance, there's a small hairpin in a particular, next to a particular weak start codon, and that affects the rate of translation. So it has biomedical implications. So the geometry of RNA viruses, first of all, you can't have RNA that's just out floating in the cell, otherwise it will be degraded. So these RNA viruses are basically packaged into typically protein capsids, and they come in different forms, and so at the upper right there's some pictures of them. But one of the most common is this quasi-icosahedral capsid. And so basically many of the RNA viruses are packaged essentially into an icosahedron. And it's not known why they do that, nor is it known how that happens. But one of the things you might recognize is that an icosahedron is one of the 5 fundamental Platonic solids. So that's the from Plato to Pasteur connection. So from Platonic solids to viruses. And of course, there's the tetrahedron, the hexahedron, the octahedron, the dodecahedron, and the icosahedron. If I look at them as planar graphs, that is, just squash them down in the plane, that's the top row that you see there. And then, of course, there are dualities. So the tetrahedron is self-dual. The hexahedron, or these cube, more commonly known, and the octahedron are dual, as well as the dodecahedron and the icosahedron. And this duality will come up in just a minute. But first, every talk should have at least one proof in it. So this is the one for this talk. Why 5? So one of the things that I find the most appealing about mathematics is that when you ask questions, there are answers to them. So my father is actually a mathematician, and one of the corollaries to this is that I went on sabbatical when I was 9. So when I was 9, we were outside of Rome on a beach, and I asked him, why do we count by 10? And he held up 2 hands. And then he picked up a stick, and he started proceeding to explain to me how we would count if we had only 2 fingers. So at the age of 9, on a beach outside of Rome, I learned how to I can't think of the words have failed me, the correspondence between binary and decimal numbers and how to convert between the 2 of them. But similarly, you might ask for the Platonic solids, why are there 5 solids? And that can actually be answered using Euler's theorem. So Euler's theorem, if we're thinking about planar graphs, tells us that the number of vertices minus the number of edges plus the number of faces should be equal to 2. And
if we're thinking about platonic solids, then they are regular polyhedra. And so that means that they have that all of their vertices have the same degree, and all of their faces have the same number of edges. Okay, well, we're going to use those two facts, and also the fact that so each edge connects to two vertices. And so this is the total number of vertices, and each edge connects to two of them. Each edge also is the boundary of two faces. So these are the total number of edges around the different faces, and I have to multiply by twice because of that thing. And then I just solve for E and plug that into this equation, do a little algebra, and then basically observe that this numerator here, it has to be greater than 0. This numerator here is greater than 0. So that means if I multiply by -1 and add 4 to both sides, this value has to be less than 4. And if I factor that, then I get these two numbers, which are both integers, have to be less than 4. And there are exactly 5 combinations of those. And so these 5 combinations, these m minus 2, this is 2 less than the number of edges around every face. So that if m minus 2 is 1, that means that all my faces are triangles. And if n minus 2 is 1, then all my vertices have degree 3. And that then gives me the tetrahedron. Okay. All right, so if we're thinking about icosahedrons and dodecahedrons, then when I got to Georgia Tech in 2006, I met my faculty mentor, who is a structural biophysicist, and he took me out to lunch. He said, "How are things going, and how are you settling in?" And I said, "Fine." And he said, "Great, because we have science to discuss." I said, "Excellent." So he actually builds three-dimensional structures of viruses, and he had been working on this virus, it's a pericottavirus, and this is actual experimental data. This is cryo-EM data. And what you see around here in the purple boundary is the protein capsid, and the gold interior is the RNA genome. Now, when you get cryo-EM data, it's very noisy. So in order to get a signal, they have to do averaging, which means that the only thing that you're going to see in this, the only thing that you're going to see are things that are invariant under the icosahedral averaging of the cryo-EM data. And the dodecahedron is. So because the dodecahedron and the icosahedron are dual, then when we do the icosahedral averaging, you see this dodecahedral cage pop up. And one could then ask, how is it that this RNA viral genome is folding into this dodecahedral cage? And this is, in fact, a picture from a Journal of Virology paper that Steve was a co-author on. And he told me they were very excited about the fact that they could take this graph and trace out basically, so each edge in this graph is covered by 2 red lines, one going one direction and one going the other direction. And then these red dots are the places where the virus sort of drops down into the center of the capsid and returns back out to it. And he was really excited about the fact that they had been able to find such a path in the graph in a matter of minutes. Now, those of you who think more combinatorially, as I do, might be starting to repress a smile or a snicker or just starting to laugh and point at your potential collaborator, because what they have done is basically realize that they have found an Euler tour in this graph. So every vertex has even degree, which means that basically if you just set off on a random walk on this graph, then you're pretty much going to end up finding one of these Euler tours. If you don't, you just patch like subcycles together. So the fact that they were able to do this is it's easy because of this Euler tour property. And it also means there are a very large number, like on the order of billions, of different possibilities in these paths. So the fact that there exists one is not going to help you trying to figure out how this RNA how this sequence is folding into it. And so this is a very nice, or one of the evidence that this is a nice biomath problem, is that I got funding from through a joint NSF-NIH panel, and ended up getting an NIH R01 that funded this project with Steve Harvey as one collaborator and David Bader, who brought high-performance computing expertise to the table. So why is this hard? Well, basically, the slide that I showed you before had this nice kind of pipeline where you have the primary sequence, and then you have the base pairings of the secondary structure, and then it folds into this three-dimensional structure. But that first step, is very, this arrow is very undetermined. So with this particular viral genome, I have a sequence, actually I have two sequences, slightly, just to make it a little bit more complicated, one of which has about 3,000 bases and one of which has about 1,300. And there are, without exaggerating, to say hundreds of different possible secondary structures with free energy near the minimum. And so you really have a needle in a haystack problem here in terms of trying to figure out what the correct secondary structure is. So the reason for that is that there are, given a sequence, even one that's as short as this is 76 bases, our tRNA sequence, there are many possible sets of base pairs. So we can really think about that sequence to structure mapping as really a one-to-many mapping, and that's highly problematic. So what do we do? Well, typically we then try to predict a secondary structure that has minimal free energy under our current nearest neighbor thermodynamic model. So without going into the details of that, basically runs of stacked base pairs are favorable because those form what are called helices, and that's a good thing. That's stabilizing the structure. Loops are the single-stranded regions that exist because this is just one contiguous sequence, and those are destabilizing. And so this optimization is trying to maximize sort of the number of stacked base pairs while minimizing the cost of the loops. And dynamic programming can solve that efficiently, but it's long been recognized that this objective function is ill-conditioned. And what does that mean? Well, it means that there are many, so you can predict an optimal structure under this nearest neighbor thermodynamic model, but there are many significantly different structures that are within a very small energy increment of the optimum. And so just to give you an illustration of that, here are three possible secondary structures for one, actually both of them concatenated of those RNA viruses, viral genomes. And you can see they're really quite different. Yet their free energies differ by 1%. So but well below kind of the margin of error in our optimization function. Given this, one can ask questions like, okay, so it's really hard to predict the secondary structure, but I might settle for just tell me what are some, like a collection of likely helices. So what characterizes strongly encoded, likely functional, and therefore existing in the native structure helices. Another question that one might ask is, all right, branching seems to be making a difference. So for instance, these are two sequences. This is the shorter of the two genome sequences, so 1,300 bases, and this is a predicted minimum free energy structure. And then the sequence has been scrambled and run through the same prediction algorithm, and so this is the MFE prediction for that one. And the biologist, or the structural biophysicist, looks at this and says, well, these two are, sort of this obviously doesn't look natural to me. It looks different than this one, and I would like to be able to characterize that. So questions that one could ask then are, what are the trade-offs among different types of loop structures? And is there a typical degree of loop branching? And then the way that I thought of doing that is, well, let's take these RNA secondary structures and let's model them as trees. So they're rooted trees, and because the ends of the sequence occur here, the root is actually at the bottom of the tree, whose subtrees are all linearly ordered. So you're just mapping the stacked base pairs over to edges and the single-stranded regions to vertices. These are actually very well-known types of trees. So they're called a plane tree, also variously in the literature ordered or linear trees. And they're rooted trees whose subtrees at any vertex are linearly ordered. And that just means, well, first of all, the roots are here. But that means that because the subtrees are linearly ordered, this tree is different than this one, because it matters that this child comes first and that second, whereas this here is first and that is second. And these are one of the many sets of combinatorial objects that are enumerated by the sequence of Catalan numbers. So I think Stanley has upwards of like 140 different instantiations of this sequence on his webpage. So it's a really sort of well-studied class of combinatorial objects. And then we can think about, but if we're thinking about these trees as sort of modeling RNA secondary structures, then there are, as you might hear, so these are the five secondary structures for this combinatorial sequence, where I basically just have A, G's, and C's. And so I have nice runs of CG base pairs and nice loop regions that have these poly A segments. And these correspond exactly to the five trees that you saw on the previous slide. And what's interesting about this, and this is sort of the math as motivated by biology part of it, is that you can think about moving around among these trees by exchanging the base pairing between different runs of C's and G's. And that's not something that that naturally came up in combinatorics before. And so thinking about the trees as these, in terms of these RNA secondary structures, it's an obvious thing to think about exchanging the pairing between them. And based on that, then you can define this combinatorial optimization. And that actually then gives results on enumeration results both on Kuros complementation and also on meander graphs that were previously unknown. So this is an example of how biology can motivate. So biology motivates new mathematics. And this is the graph that you get if you move among those five RNA secondary structures with this helix exchange operation. But it's also important that mathematics yields biological insights. And based on this tree model for the RNA secondary structures, it's possible to prove basically that local helical constraints are necessary and sufficient for the folding of global structure. That means if I want to control the overall structure of this RNA molecule, I obviously have to worry about what's going on locally. And in the combinatorial model, that is both necessary but also sufficient. And that's an interesting idea, because that then suggests that this question about well-determined viral structures, maybe you want to look at the distance of the base pairing, and that's sort of a key consideration in terms of the finding well-determined helices. Another result that you can prove is that associated loop energies are minimized by maximizing vertices with free edges. So basically, in this thermodynamic model, branching is locally favorable but kind of globally balanced. And again, that was something that people hadn't really been looking at before. And so it's interesting that you can prove that in this model of RNA folding. And then that, if you take that then and try to think about what that's telling you about the biology, well, that's suggesting that the branching degree in these viruses may make a significant difference. And then beyond these results, so I have a probabilist colleague, and he got interested in this problem. And so we were able to basically characterize the typical branching configurations by giving a large deviation principle for the branching of these trees under the current thermodynamic model. One of the things that's nice about that is that it's simple enough. This result is sort of, we can ask how the branching changes under different thermodynamic models, because the model has been evolving. This was an older version. This is sort of the current version. And you can see from this picture that there are some significant differences between those two versions. So they really, a major change has been this change in terms of the favorability at low degree branching. And then the tails actually decay at different rates. So that is another thing that's important. You can also look at, you can use techniques from geometric combinatorics to look at the parametric analysis of RNA branching. So here I have theorem, and then I have a picture. So you'll forgive me, but what this picture is it's actually it's a polytope, and it's basically it's the cutting out the parts of the parameter space, and the vertices correspond to these extreme cases. So basically, there are two extreme cases that have the high degree of branching, where branching is sort of the most favored. Another extreme case is where branching is essentially unfavored, and then this resembles, of course, the canonical DNA double-stranded helix. And the regime that the current model exists in is sort of close to this, where branching is favorable, but locally, as I told you, but kind of globally balanced. So we don't have we don't have tree structures that have really high degree of branching, unless possibly that there's some functional significance to that. So back to our RNA virus. One of the things that came out of this is this idea, as I told you, that somehow locality is important. And previously, this model of base pairing had been taking what's called the backbone of the virus. So this had been one contiguous run of the sequence where sort of the fingers drop down into the center of the molecule. And I said to my collaborator, is there any reason why it should be that? So he started calling this the stalactite model because they're dropping down into the middle, as opposed to the stalagmite model because they're growing up from that. So is there any reason to believe it's this instead of that? And he said, now that you mention it, no. So the idea that this dodecahedral cage is actually built out of discontiguous pieces opened up new possibilities and new ways of piecing together this RNA viral genome. And so this picture was actually produced by Steve's graduate student, Ying Zheng, because she found a way actually of doing that. So she took, this is our dodecahedron that we want to cover. She took a predicted secondary structure from RNA, from the longer sequence, and basically identified regions of it that she could then piece together in a reasonable way to form this dodecahedral cage. And so that's really, I think, neat. So, in summary, this is a picture of Ying so biologists still build actual physical models of things. And I really like this picture because she's standing there, just like Watson and Crick with their DNA model. She's got her RNA dodecahedral model where she's trying to figure out how to fit the helices all together. And really what I hope, if you take away only one thing from this talk, it's the idea that the interaction of mathematics with biology has many facets. And some of them are less well known than others. And the one that I work at is this interaction between discrete mathematics and molecular biology. And there's a lot of interest there. So 2 final thoughts. First, this is one Harris comic. He's, you would recognize these columns. It's and you need to be more precise in step 2 comic, or when you have the mathematicians at the blackboard and then a miracle occurs. He's that comic. But this is the megatrends in science. So particle accelerator to circle Australia. Math and physics. The interaction of math and physics is well known to be both beneficial for physics as well as very fruitful for mathematics. Chip, now capable of infinite number of computations per second. Again, the interaction between computer science and math, very well known to be both beneficial for computer science as well as fruitful for mathematics. Common cold virus remains elusive. So I think, but of course I'm biased, that biology is probably the most challenging math plus X frontier. But there's a lot of potential here to be both beneficial for biology as well as fruitful for mathematics. And it's important to keep that in mind. So that's final thought 1 and final thought 2. OK, thank you.